I'm going to pick up from where we were in the lecture, and I'm going to do 2 short videos in order to complete the material from the lecture. The first video, which is this one, is going to be dealing with these 2 sections on genomic annotation, well, really ENCODE and junk DNA, and repetitive DNA. And then the second video is going to deal with genome assemblies and genome annotations. Actually, this is a bit of a mistake. It shouldn't see genome annotation here. We're going to be dealing with ENCODE and junk DNA. So let's get started. So here's a little bit of history. The Human Genome Project was started in 1990, and the draft Human Genome was published in 2001. And it was actually published by 2 different competing groups. One group was a public effort across multiple countries, led by government organizations in those countries. The other effort was a private effort by the biotechnology company Celera, which had been founded to do just this. Now, at that point, the sequence was mostly done. The draft was mostly done. But the annotation of the genome was really poorly understood. And in fact, one of the results of the Human Genome Project was that only a small percent, 1% of what was sequenced turned out to be genes. We've seen this earlier when we were discussing the C-value paradox, that protein coding genes are just a tiny, tiny fraction of what's contained within the human genome. And it's fair to say that the number of protein coding genes was a big disappointment to many people working on or waiting for the completion of the Human Genome Project. At the time, there were actually bets. There were bets in the field about how many human protein coding genes there were. And most of the bets were north of 100,000 genes. And then it turned out that the number was really small. It was basically 20,000. And this was a huge surprise to many people. And actually, to a lot of people, it was a great disappointment. We would like to think that we would have more genes than some other organisms, which seem at first blush much less complex. But like the C-value paradox, it's not just the size of the genome, it's actually the number of genes also has a bit of a paradox to it. Well, what that meant was that with all the promise of the human genome, only 1% of the human genome was known to be genes, and the rest was something else, right? So what was it? Well, one major possibility, was that a lot of the rest of the genome, or some large fraction of the rest of the genome, was regulatory, meaning it contained information in promoters, in enhancers, and in other regulatory elements that actually controlled the expression of those protein coding genes. So maybe there was information that was missing about these regulatory elements in the genome, and maybe we can try to find that. And so there was a major project launched in 2003 called ENCODE, and it's still running today, to try to address this. And you can think of this as kind of coming out of both the success and the disappointment of the Human Genome Project. Success in that, well, the Human Genome Project was done, but disappointment in that at that point, most of the genome appeared dark and possibly useless, and that was embarrassing to the people who put so much money behind it. So ENCODE is this project to map those things that are not just the genes themselves, although it also helps to fine map the gene, but it's a map of the structure of the genome. And a lot of ENCODE is done by mapping where certain kinds of structural elements occur, or functional elements occur. Now, many of the techniques used in ENCODE rely on massive sequencing. And these sequencing efforts do things like interrogating where in the chromatin the genome is, say, more open. That's this part right up here, right? So chromatin is, in eukaryotes, is packed, and at some places the chromatin is more open, and at some places the chromatin is more tightly wound up. And those places where chromatin is more open are more accessible to transcription factors and other DNA binding proteins, and so they're more accessible to regulation. So knowing where those regions are can be informative, and these regions are found by doing sequencing. But the sequencing is done using a preparation. So the preparation is different. So the preparation will be done in such a way that they are enriched for open chromatin by, for example, hypersensitivity to endonucleases. The sequencing is massive, and the sequencing is unbiased. But the library preparation methods will capture the DNA with certain properties, like here at the positions that are hypersensitive to endonucleases, and by that we infer that the chromatin is more open. Another method here is called ChIP-seq, and in ChIP-seq, histones or transcription factors that bind to sequence-specific, have some sequence specificity, are immunoprecipitated on chromatin, and then they bring the DNA down with them. And when that happens, those elements, those attached pieces of DNA are sequenced, and we can identify where those sequences are in the genome. And that tells us where the transcription factors are bound or where those histone modifications occur. Another one is labeled here with this CH3. And that's an epigenetic modification of DNA methylation, which occurs on many cytosine residues. And then this other one over here, labeled 5C, it's labeled with that simple two-character label, but it actually refers to a whole bunch of techniques that help to identify medium and long-range interactions between different locations in the chromatin that come together to form organized loops. All of these data are available in genome browsers as additional tracks that tell us things about where regulations occur and what kind of regulation might be occurring at different places. That's the whole effort. That's the effort of the ENCODE project. So the main goal here is to create this comprehensive catalog of functional elements in the human genome. And the functional elements are those elements that control the function of genes, the regulation, the expression of genes. Now, in ENCODE, most of the technology to generate this catalog didn't exist when the project was started. So quite a bit of it was about technology development. And the first phase, the pilot phase, was to create the approaches to identify those regulatory elements. And then that was followed by the development phase, which was to develop the technology to scale that up so that those newly invented methods could be applied. And then after that is production, which is doing it in earnest and analyzing that data, and that's where we are now. Now, at this point, thousands and thousands of experiments have been performed, and a lot of the results can be incorporated into genome browsers. Most of these experiments are performed in cell lines, and cell lines function, of course, depends on cellular context. Right? So a hypersensitive site, a hypersensitive site that might be observed in one cell line may not be a hypersensitive site in another cell line because the chromatin might be more packed up in that other cell line. So it's not like the original genome project where there's a reference for the genome which is the same in every cell of an organism. But in this case, where you're looking at these structural elements or these epigenetic elements, the functional elements, many of them may be common, but many of them may be different depending on how they're measured. So there's a lot of annotation, and that annotation is done in different cell lines so that they can be layered on top of each other, like promoters, enhancers, and silencers, transcription factor binding sites, and repetitive elements. The first ENCODE papers were all published together, and there were a whole bunch of them published together. And they were published from a bunch of different groups, and there was a big hoo-ha about it. And one of the big findings that got a lot of attention was this first bullet point, that the majority of the human genome is transcribed, undergoing active transcription. Now that seems weird, right? We just said that only 1% of the genome is actually genes. So what's that about? Why is the majority transcribed? And the second point is that the second conclusion was they said that 80.4% of the genome is functionally active. And then if they exclude RNA elements and histone elements, then 44% is what they call functional. They also identified a bunch of non-coding transcripts, which are important, really important, and they identified and mapped many transcription start sites, regulatory sequences, and so on. But these first two bullet points are really controversial, to say the least. Okay, so there was a lot of critique of these bullet points. And it kind of relates to what I said earlier, that if one thinks of the ENCODE project as coming from, to some extent, disappointment in the amount that we learned from the original Human Genome Project, maybe some of these conclusions are attempt to rescue that. If the majority of the human genome is transcribed and 80.4% of the human genome is functionally active, does that mean that junk DNA is dead, is not a thing? Well, what does functional mean is maybe one thing here. So there was a lot of critique of ENCODE, and especially because this part here, 80.4% of the human genome is functionally active, seems in conflict with this 5% of the genome is under evolutionary pressure in mammals. So there were a lot of critiques, and the thrust of most of these critiques was that ENCODE's definition of functional was too broad to be biologically meaningful. And they kind of said function means it does something, like being transcribed. But function has to be reflected in evolutionary conservation. Functional sites should be transcribed. Just because it's making something doesn't mean that that's actually functional. Another point is that while selfish repetitive elements like transposons, they have ENCODE-defined function like transcription, but they don't really have any biological function. And if they did have biological function, then the ENCODE authors wouldn't need to make up a new definition of function in order to ascribe function to this. So there were a lot of critiques of the ENCODE results in that way, especially their treatment of the idea of function and the notion of junk DNA. There are lots of critiques worth reading. Two of the best critiques, I think, are these critiques by Sean Eddy in 2012 and Doolittle in 2013. Both really good, and they both address some of the issues that I mentioned here. But the issue between the evolutionary evidence and the ENCODE's definition of function seems to be kind of the main thrust of these critiques. Okay, let's switch to repetitive DNA. So how much of the genome is composed of repetitive elements? In prokaryotes, it's actually quite small, right? Very small, some maybe even almost none. But in Homo sapiens, it's over 50% of the genome is composed of some form of repetitive elements. And this is from a paper, Richard, Karras, and Dujon, which describes what types of repetitive elements there are. And what we can do is take a genome, we can ask and take a genome that we're interested in, we can just take it and let's take out the unique DNA sequences and then ask questions of the remainder. We can start asking, well, what are these like? Okay, so we have repeated DNA sequences. Over here are repeated DNA sequences on the left that are kind of pulled together in tandem, lots of tandem things, and then on the right are these ones that seem to be in different places. If we look just at the tandem repeats, which might arise by gene conversion or replication slippage, we have things like tandem paralogs, where you have paralogs that appear right next to each other. We have what's called satellite DNA, which comes in these various flavors, as well as ribosomal DNA. So ribosomal DNA tends to occur in these arrays, these tandem arrays. And then over on the right side, there are these dispersed repeats, which include a whole other class of repeats. But if you look at them, they actually rely very heavily on transposition and retrotransposition. That's what these RTRs are, and the TRA is transposition. And they include paralogs. Okay, we saw paralogs here, so there's another type of paralog here. This is a paralog that arises by retrotransposition. And then transposition, and then tDNAs, which are elevated copy numbers of tRNA copies. And we can divide the transposon elements down here into different types. These are the RNA-derived ones on the left, these three on the left, and then the DNA transposons. Oh, okay. Now, repeat sequences, they can be detected by computer programs designed to do this. There's one a popular one called repeat masker, and you can basically screen a DNA sequence or a whole genome for interspersed repeats and low complexity DNA sequences. And by repeat masker measures, 56% of the human genome sequence is considered repetitive. And you can mask that in analysis. So now let's, actually there's a website here too. Now a little bit about the repeats, in particular the repeats on the right side, those dispersed repeats, these transposon-derived repeats. How are these generated? Well, a lot of them are generated by copying RNA intermediates, and that comes in as a retrotransposon, or DNA elements, which are DNA transposons. In the RNA, there are these subclasses, these LTRs, which are long terminal repeats. These look a lot like retroviruses. And these lines, which are these long interspersed repeats, these actually encode an enzyme that has a reverse transcriptase activity. And then the signs, which are also RNA mediated, and these include ALU repeats, which you may have run across. But look up here at the second bullet point. If an mRNA is reverse transcribed and then integrated into the genome by retrotransposition, it'll look like the gene, but it will lack its introns, and it'll have these short flanking repeats from the retrotransposition insertion. And then, of course, there are DNA transposons on the bottom of this. So here's an example of a pseudogene that's derived by retrotransposition. So this is the gene ADAM20 and the pseudogene ADAM20 and the gene ADAM9. So ADAM9 is an ordinary gene. Okay, so this is an ordinary gene. We can see it's got four transcript isoforms here, and these long introns, and you can probably recognize that this layout is from the UCSC genome browser. So it has lots and lots and lots of introns and some small exons. But on the bottom is ADAM20. So ADAM20 is on a different chromosome, and it looks like just this two exons, one intron here. And notice the repeat sequences that occur in the introns in both cases. It's more obvious that they're in the introns in ADAM20 because we can see them. And so how did this thing arise? Well, it looked like it arose less than 20 million years ago because it occurs in human and chimpanzee, but not in macaque, which diverged from a common ancestor 20 million years ago. But probably more than 5 million years ago when humans and chimpanzees diverged from a common ancestor. So it's an mRNA that's been retrotransposed and formed a pseudogene. And then afterwards, probably these repetitive elements, the lines and the signs and the LTRs, entered in and gave us this new intron structure. Okay. This type of pseudogene origin is not uncommon, and it's referred to as a processed pseudogene. There's a lot of retrotransposition from mRNA, and most of the time it won't lead to expression, and that's because the promoter is missing. Right? This is an mRNA, the promoter is missing, and unless it happens to land in a place where there's a promoter just lying around, it's not going to be expressed. And instead, what happens, it'll be this thing that looks like a gene, maybe even has a poly-A tail in the DNA, and it will show up in the chromosome wherever it lands as this processed pseudogene. That means that there are two classes of pseudogenes at the highest level. There are these processed pseudogenes, which lack introns, and we see them as arising from retrotransposition of mRNAs, as just described, and the unprocessed pseudogenes that contain introns, and they're produced by the gene duplication mechanisms that we discussed earlier, like unequal crossing over or replication slippage. Pseudogene annotation tracks are available in the genome browsers. You can download them from Ensemble, from the UCSC table browser, and from this website. Another class of repeats are these simple sequence repeats like microsatellites and midi-satellites. Microsatellites are these very, very short sequences that are repeated many, many times. And mini-satellites are a little bit longer, so maybe 1,000 to 500 base pairs. And they're still relatively simple repeats. And the thing is, they can expand by replication slippage. And in human, for example, there's a triplet repeat, CAG, which is, when it expands, it seems to occur in a lot of different genetic disorders like Huntington's disease. And the expansion of this kind of triplet repeat is one way to identify a gene that is associated with a complex phenotype, which is what you're looking at in CA2. So here's an example of some simple sequence repeats in the UCSC genome browser, the lines, the signs, the LTRs, and you can see them interspersed here, generally speaking, not in the exons. And these are annotated by repeat masker, and that's how they wind up in the UCSC genome browser. The next type of repeat is a segmental duplication. So this would be a longer region that for some reason duplicates, and these would be reasonably long. They can, maybe 1 kilobase to 300 kilobases, and they might get copied from one chromosome to another. They're pretty common. 5% of the human genome seems to consist of these kinds of segmental duplications. When these duplications do occur, they seem to share a very, very high sequence identity. And finally, there are these blocks of tandemly repeated sequences. And some of these sequences are functional. Not functional in the sense that a gene is functional, but functional as targets for something useful. So, for example, the human telomeres contain thousands of this repeat of TTAGGG. And that repeat in telomeres helps maintain the integrity of the ends of chromosomes. And as we age, the integrity of the ends of chromosomes is important for healthy aging. So this is part of what that does. The repeats within centromeres help in positioning spindle attachment, which is required for replication. And that's where I'm going to end today, this section, and we will go on in the next short video to the remainder of the material for this lecture.